Welcome to my YouTube video. In this video we are basically going to look at digestion and dentition in humans. So the diagram that you're looking at right now is a diagram that is showing all the digestive organs, or the organs that are involved in digestion. Remember, we learned to say when you've got a group of similar specialized cells, if we say the cells that we're talking about, a group of similar specialized cells, it gives us what is referred to as a tissue. Then we know that two or more tissues are the ones that give you what is referred to as an organ. Now today we are looking at the organs that are involved in the digestive system, because two or more organs performing different functions, they are going to form what is known as a system. So in this case we are basically looking at a system that is referred to as the digestive system. So this is the digestive system. So the digestive system, we want to take a look at which organs are really involved in the digestive system. So I've labeled a few. We have got A, which is basically the what? This is the mouth. So a lot of things happen in the mouth. Number one, we know that the type of nutrition that happens in this tube that is running from the mouth and it goes all the way to the anus, the type of nutrition that happens there is referred to as holozoic nutrition. The type of nutrition that happens in such a tube, let me just erase here so that I explain here. The type of nutrition that happens in such a tissue is referred to as holozoic nutrition. Yes, holozoic nutrition. So holozoic nutrition is a type of nutrition that happens in an animal, and if we were to classify this nutrition, it is falling under heterotrophic nutrition, where an organism obtains nutrients that are found in other organisms, or in the body of another organism, and we are examples of heterotrophs. And we carry out holozoic nutrition, which is happening in this specialized tube known as the alimentary canal, which begins from the mouth, and it involves 5 stages. And the first stage is basically the taking in of food into the mouth, and it is referred to as ingestion. Ingestion is basically the taking in of food into the mouth. Okay, now food has landed into the mouth. What is it that happens in the mouth? So within the mouth, that's where we're going to have what is referred to as that's where digestion begins. What type of digestion, to be specific? That is physical digestion. Why are we calling it physical digestion? We're calling it physical digestion because we are physically breaking down the large food particles into smaller ones. Exactly. So if you have eaten food containing starch, if you've eaten a carbohydrate, food containing starch, so this food containing starch, there is this guy labeled B. This guy labeled B is the one we are calling salivary gland. So the salivary gland, let me just write it well. The salivary gland is a gland that secretes enzymes. In this case, we're talking about the enzyme called salivary amylase. We're calling the enzyme salivary amylase. Salivary amylase, what does it do? Salivary amylase, it basically breaks down starch into maltose. and that we can call that one a bit more of chemical digestion, because an enzyme is involved in the breaking down of large food molecules into smaller ones. So I'm saying in the mouth we have got digestion. What type of digestion? Physical digestion, because it involves the breaking down of large food molecules into smaller ones physically, and this is done with the help of the teeth. But again, we have realized that there is an enzyme from the salivary gland known as salivary amylase, which is breaking down starch into maltose. So you can say in the mouth we have a bit more of chemical digestion as well. You might agree with me on that one. For chemical digestion as well. So this, to summarize everything, we are saying we have got two types of digestion. Digestion can either be physically, called physical digestion, or it can be with the help of enzymes, called chemical digestion. What type of food is digested in the mouth? We are calling it, what type of food is digested in the mouth chemically, or chemically, if you to specify, that is starch. With the help of what enzyme? The enzyme is salivary amylase. I think we can proceed from there. We can now go straight to C. What is C? What is C trying to talk about? What is C? So C, if you look at C, C is basically the esophagus. C is the esophagus. Esophagus. That's where you're going to observe the wavy-like motion of food. That's called peristalsis. That's where peristalsis occurs. Apart from that, you can also find peristalsis in the small intestine. That's one of the regions of the digestive system where peristalsis occurs. That is one of the regions where peristalsis occurs. Where peristalsis occurs. Yeah, that's one of the regions where peristalsis occurs. Apart from that, we can now proceed to an organ known as E. I think on an organ E, we're going to dwell there a little bit more. So E is basically what is referred to as the stomach. So when you look at the stomach is in simpler terms, we normally say when you're looking at the stomach, we normally say that the stomach is an elastic bag with muscular walls. So it is an elastic bag. It is an elastic bag. Elastic bag with muscular walls. Muscular walls. Yes. So that's what the stomach is. It's basically an elastic bag. Elastic bag meaning it can stretch. It's an elastic bag with muscular walls, meaning it has got muscles. Its walls is made up of muscles. But there's something that is very, very important that is produced by the stomach, the walls of the stomach lining. So the walls of the stomach lining produce what is referred to as let me write it down. What is referred to as gastric juice. So they can ask you a question to say, where is gastric juice produced? The answer is the stomach. Where is gastric juice produced? The stomach. So what are the contents of the gastric juice? So you realize that the gastric juice that is being produced is basically a mixture. So gastric juice, we are saying, you can write this down, make sure you say right. Gastric juice is a mixture, let me write it properly, is a mixture of pepsin, renin and hydrochloric acid. So we've got hydrochloric acid, we've got pepsin, and we've got renin. So that's what makes up what is referred to what. That's what, and basically there should be another one, and mucus, the last one, mucus. So I mean the composition of gastric juice that is produced by the stomach, it has pepsin, renin, hydrochloric acid, and mucus. Now let's try to break it down. Let's try to talk about the function of each component. I think let me start with hydrochloric acid. What is the function of hydrochloric acid? Let me write it over here. Hydrochloric acid. So hydrochloric acid present in gastric juice, what it does, number one, is the simplest, is it kills the microbes that might be present in the food. So if you eat your food, and if there are any microorganisms like pathogens, we can talk about bacteria and these germs. In simpler words, people are too used to the word germs. Okay, so let me say microorganisms or microorganisms that may cause harm to the body. The microbes, it can be bacteria, fungi, name them. Those microorganisms, if they are present in food, they might be eliminated by hydrochloric acid. So we can just put it to say it kills microbes that may be present in food. Or let me just say present in food. Let me just say that might be present in food. Yeah, that makes sense. That might be present in food. That is one. Then two, I want you to understand to say pepsin. When you look at pepsin is the active form. It's the active form of pepsinogen. So you need to understand that the enzyme pepsin is secreted in its inactive form called pepsinogen. Now it is because of the presence of hydrochloric acid that is released, where pepsinogen is converted to pepsin. So you can say hydrochloric acid is important in the activation of pepsinogen to pepsin. So it's because of HCl. That's one of the functions. So we've got 2. 3, we can also say this one is the one that sets up the acidic pH. The acidic pH at which enzymes of the stomach work best. Enzymes of the stomach work best. Yeah, I repeat, you can just write that down so that it's nice, so that your work is flowing. So we are saying number 1, hydrochloric acid kills bacteria or microbes. It kills microbes that might be present in food, and this may include bacteria, fungi, and other microorganisms that might harm the body. Then we also have pepsinogen being converted to pepsin. So this is facilitated by hydrochloric acid. Then apart from that, hydrochloric acid sets up the acidic pH at which the enzymes of the stomach work best. So it's more like I'm telling you to say pepsin, which is a protein digestive enzyme, works best at acidic conditions. It works best in acidic conditions. What type of food is digested in the stomach? What type of food is digested in the stomach? Type of food? We've got proteins. Here proteins are broken down into peptides. Proteins are broken down into peptides by pepsin. and of course this entire process is happening in the presence of hydrochloric acid. All right, let's try to talk about the function of yeah, I think that's nice, that's important. Just need to notice to say gastric juice produced by the stomach, it's a mixture of pepsin, renin, hydrochloric acid, mucus. And for the rest of the points, renin, mucus, just contact me at 0970865597 if you want more information regarding the detailed information on this stuff. Then we can proceed to the other guy, H, which is the pancreas. Let me just erase this part of the board. Erase this part of the board. Yes. Alright, so now we can proceed to this guy called the pancreas, which is basically H. So H is the pancreas. H is the pancreas. What's the function of the pancreas? So the pancreas in digestion, what we need to understand is it's going to secrete what is known as pancreatic juice. So the pancreas secretes what is referred to as pancreatic juice. So this pancreatic juice, it's also a mixture. It's also a mixture. So when you're looking at pancreatic juice, one thing that you're supposed to understand is that it's produced by the pancreas, then it flows through this duct. This duct that I'm talking about here, it's the labeled I. It's also known as it's the pancreatic duct. So this is known as the pancreatic duct. So after pancreatic juice is produced, it flows through the pancreatic duct all the way to this region, which is known as the duodenum. Yeah, so the pancreatic duct to all the way, not until it reaches the duodenum. Now after that, what is it that you're supposed to pay attention to? What is it that is all about this juice that you're supposed to pay attention to? So we're saying pancreatic juice contains it contains sodium hydrogen carbonate. It contains sodium hydrogen carbonate. Sodium hydrogen carbonate. Guys, this is an alkaline substance. It's an alkaline substance. Remember, we might have a food bolus that is coming from the stomach. It has already been worked on by pepsin. In case of any protein in it, then it is coming from an acidic environment. Then boom, it's coming. It has reached here. It's now in the duodenum. So it carries that acidic chyme in it. It carries an acidic chyme, according to, I don't know how you can spell it, but it carries this acidic chyme because it's coming from an acidic environment. But now it's going in an environment where for the enzymes to work best in the duodenum, the environment has to be alkaline. That's the reason why in pancreatic juice you're going to find this guy known as sodium hydrogen carbonate. Sodium hydrogen carbonate. This one, it neutralizes the acidic chyme to alkaline. That's one of the things that you're supposed to know. Then secondly, you're also supposed to understand to say within pancreatic juice, we have got three enzymes that are present in pancreatic juice. Number one, we have got what is known as pancreatic trypsin. We've got what is known as pancreatic trypsin. What does pancreatic trypsin do? We'll talk about it. So we've got, let me just erase a bit here. So we have got sodium hydrogen carbonate 2, pancreatic trypsin, 3. We've got pancreatic lipase. So since proteins have come, the trypsin is going to finish up the proteins. Then the lipase is the one that now digests the lipids, breaking it down into glycerol as well as fatty acids, which are basically the building blocks of lipids. So if you've got lipids like this, what happens is pancreatic lipase, this is the enzyme that's going to digest the lipids into its constituents, like into its basic units. So in this case, I've got this how it is shown in most of the books. And that guy. So just know that these are the fatty acids. So I've got 3 fatty acids, then we've got 1 glycerol molecule. This is a glycerol. So in the duodenum, lipids are going to be broken down or digested by the enzyme known as lipase into its building blocks. In this case, we're talking about glycerol and fatty acids. How many fatty acids? 3. How many glycerol molecules? 1. So if the diagram, I want to take a look at the diagram properly, it looks something like this. Something like that. Pay attention to it. Most of the times the examiners might just bring that diagram and they'll ask you to label. So it looks something like this, something like that. So this is a fatty acid. This is a fatty acid. Also this one, also that one. So there are 3 fatty acid molecules. Then this guy here, this one here, if I change the color, this entire thing, this, that is a glycerol molecule. Basically that's what happens. Yes. Then what else is found there? We have got pancreatic maltase. Apart from lipase, apart from trypsin, the third one is pancreatic maltase. Remember that maltase is an enzyme that is going to break down maltose into glucose. That's something that you need to understand. So that is happening in the duodenum. So we are saying here we've got trypsin, we've got lipase. I've talked about what lipase does. Then we've got maltase. What does maltase do? It breaks down maltose into glucose. Basically that's it. Yes. So now there is another question that has been asked here. There's another question that has been asked. I don't know if you're able to see it, but if you're not able to see it, let me just clear the path so that you can take a look at it and write it down. So I'd love you guys to write down this question, just to summarize up, because we've got a lot of work. The question is, which of the organs above carries out deamination? So guys, deamination happens in the liver, and deamination, in simpler terms, we're just basically saying deamination is a process by which an amine group is removed from an amino acid. It's just basically the removal of an amine group. Let me write it. So deamination is the removal of an amine group from an amino acid. From an amino acid. Then at the end of the day, what we find, we end up making what is referred to as urea. So urea is less toxic as compared to the excess amino acids. So urea is less toxic as compared to the amino groups. So because we have removed the amine group from amino acid, we are going to form something that is less toxic known as ammonia. So ammonia is then reduced to urea. Urea is then now the one that is safe to be taken to the kidneys and excreted. So the process is pretty simple. Just know that amino group, when you remove amino group, you produce what is known as ammonia. Then ammonia is the one that is now converted to urea at the end of the day, which is excreted by the kidneys. Then where is gastric juice produced? We have already established the fact that the gastric juice is produced in the stomach. The gastric juice is produced in the stomach. Then where is pancreatic juice produced? Pancreatic juice is produced in the pancreas. Then the other question is bile. Where is bile produced? Where is bile produced? So bile is produced by the liver. It is produced by the liver. It is produced by the liver. It is produced by the liver. Then it is stored. It is stored in the gallbladder. It is stored in the gallbladder. Stored in the gallbladder. So it's just temporarily stored in the gallbladder. But you need to understand one simple thing that I can end with here. It is the composition of bile. So bile is composed of what is known as bile salts. The question is, what is the function of bile salts? Bile salts are the ones now that break down large fat droplets into smaller ones, so that the enzyme lipase can easily work on the lipid molecules. So I'm going to explain that better when it comes to the question that is just down there, where they are asking you to explain the function of the liver in digestion. So I've got two questions. It's either they're asking you to explain or to describe. So now I can also teach you how to explain as well as how to describe. So apart from bile salts, we also have got sodium hydrogen carbonate. The function is the same sodium. It's the same sodium hydrogen carbonate that was also produced by the pancreas, which is neutralized in the acidic chyme. Sodium hydrogen carbonate. Then the other one is bile pigments. The other one is bile pigments. What's the function of bile pigments? These are the ones that add color to the feces. Then dentition. Dentition, I think in the next video, that's why we're going to explain what or the function of the liver in digestion and dentition. Thank you very much for watching my YouTube video. If you want more information, online lessons and other extra lessons, you can contact me on 0970 86 55 97. We offer mathematics, which can be pure algebra. We can also have got we can also offer pure chemistry, pure physics, and also biology, including the IGCSEs.